Sabine Hazan all the way from California, where it's 10 o'clock in the evening there and it's 6 o'clock in the morning here. But Sabine, thank you for extending your day for us.
Of course, my pleasure. I'm a night owl anyways, so it's good.
Okay, good. So basically, tell us a little bit about yourself. You're a gastroenterologist and you're researching microbiome. Before microbiome came along, what was your kind of day job?
My day job was doing clinical trials for pharmaceutical companies and bringing products to market. That was my day job. So I was the girl that was basically recruiting patients for different protocols, from C. diff to whatever. So it was interesting.
And were you doing clinical work then or as well?
I'm sorry?
Were you doing clinical work as well?
Yes, I was doing, so I started off my career as a gastroenterologist, obviously. I finished my fellowship, and my attending said, I got the Dean's Research Award at University of Florida, and my attending said, you're really good at research, you should really keep it up. And I wanted to be in the trenches, doing colonoscopies, taking care of GI bleeders, so I was like, nah, I'm just going to do clinical GI. And then, you know, 2 kids later, clinical GI was with a husband that's a cardiologist, became a little bit tough, you know, the call and taking care of the kids and juggling. So I went back, I kind of was doing 90% GI, 10% research. And then over the years with 2 kids, it became 90% research and 10% GI. And so that's been my path, essentially.
Brilliant. And when did you first hear the term microbiome?
Well, microbiome, probably at the beginning, right? Because, you know, for all of us, it was fecal material, right? At the beginning, because we were doing fecal transplant, the process of taking stools from a healthy donor and putting it in a patient with C. diff. And so, you know, probably a couple decades ago, without saying my age, but yeah, a long time ago. I remember, in fact, actually, I remember being a fellow at University of Florida and going, you know, through the posters. And my friend, Dr. Neil Stolman, said to me, the future is in SHIT. Look at the posters. They're using fecal material to fix C. diff. I mean, this is the future. And please don't make me play with poop. You know, I don't want to go there. And look at me. I'm like deep into it. So, you know, careful what you say.
Indeed. I've come across C. diff a few times. It's got a rather characteristic smell, but if you don't mind giving us a bit of background, what is C. diff?
So C. diff is a disease basically where people have severe diarrhea.
Yes, Clostridium, isn't it? It's a type of
Clostridium difficile, and by term, which is French for difficult, it's a difficult bacteria to get rid of.
Yeah.
You know, patients typically were in the hospital. However, in the last decade and a half, we started seeing a lot of young people. And one wonders, you know, were they consuming food like meat that from a cow that was that had a lot of antibiotics, and possibly that's how they got, you know, C. diff. But essentially, C. diff is this microbe that's very difficult to fix. I was the girl that was doing clinical trials to try to kill it. So when I couldn't kill it with the conventional antibiotics like vancomycin, Flagyl, and then, you know, came along fidaxomicin. You know, I did all those clinical trials, fidaxomicin, monoclonal antibodies, you know, spores. And so when you couldn't fix it with the conventional therapy, you would go into clinical trials. And then when I couldn't fix it with clinical trials, then I basically would do fecal transplant, which is basically taking stools from a spouse or a kid or a kid of a person, and then basically putting it in a person. And, you know, the field has expanded. So the beginning of it, of the microbiome really, was this discovery that you could take stools and put it in the colon and change a disease process like Clostridium difficile. I think Dr. Tom really took it to the next level of, you know, could it be useful for Alzheimer's? Could it be useful for Parkinson's? Could it be useful for autism? You know, the N1 where we basically had a diff that I was doing for transplant and had Alzheimer's, improved his Alzheimer's. So, you know, it kind of gets you into the path of seeing, well, something in the gut. What are we doing? You know, 2 cases of alopecia areata from Dr. Colleen Kelly grew after fecal transplant for C. diff. So when you see these cases, but of course nobody can refuse that, and then you kind of ask yourself, well, is it donor dependent? What's going on there? So that was essentially going after a microbe C. diff, trying to kill it. I couldn't kill it, and I couldn't succeed in saving my patient. I would do fecal transplant. And then fecal transplant, really discovering there's so much more to stools than stools, really. So it was fascinating.
So you're actually using the microbiome, basically the bacteria from someone else.
Yes.
Recolonizing the infected colon, putting the good guys in, and they kind of edge the nasty bacteria out. Is that kind of what's happening? Is it?
You know, I don't think you could say it's good guys. I think it's good guys and bad guys. I think it's a consortium of poop, which are microbes that you put in. I think, you know, the future is going to be more precise. You know, we're going to be a little bit more precise in what we put in. But right now, you know, we're at the level where we're basically taking stools from a healthy donor and putting it into the colon of a person with C. diff. What was fascinating with C. diff is that's a relatively easy condition to fix with fecal transplant. You know, you could do it via NG tube. You could give capsules, or Dr. Brody calls it crapsules. Yep. You could do via rectal. You could do via colonoscopy. And the success is pretty high, you know, above 90%. It's when you try to do it for other conditions and try to use it, it's a little bit more complicated.
But the procedure itself is basically just take some fecal material. And obviously, you screen people for infectious diseases. Presumably, you check for HIV and hepatitis. And
yeah, you have to check for a lot more. So it used to be, you know, we would just check for C. diff and E. coli and Giardia and, you know, the common parasites and microbes. But then there were a couple cases of people that died from fecal transplant. And so the FDA demanded that we look for vancomycin-resistant E. coli, carbapenem-resistant E. coli, etc. So it's a little bit more detail. The testing is very long. It's about 11 tubes of blood, and it's about 4 samples of stools just to make sure that it's clean. And so you can't just take anybody and say, oh, you know what, they look healthy, and therefore I'm just going to use their poop and that's it. No, it's pretty, you know, I'll give you an example. I'm supposed to do 2 cases tomorrow morning of people with C. diff. I was supposed to do them, and they were on my visit, and I was about to do this. And the 2 doors, and one had E. coli, the other one had Giardia. So we had to cancel cases. So, and these were people that basically not want to use the pharmaceutical product that's available. So, you know, they wanted their family members. So it becomes expensive when you do that because you have to test every single thing. And you have to test within that week period that you're going to do it to make sure that you're kind of getting the clean stools. But, you know, here we are, you know, Monday, and the 2 cases are actually canceled because, unfortunately, it's not that easy.
But presumably, if you're in, say, a spouse situation or a parent-child situation, you're sharing your bug all the time, aren't you?
Yes. But you have to remember the person that has C. diff is immunosuppressed, right? So are you giving a microbe where one spouse is strong and resilient to another one, and that's immunosuppressed? And are you going to potentially, you know, harm that person? So I think it's important to test. It's important to be very careful. You know, to me, in my lab, we go above and beyond. You know, we have a genetic sequencing lab where we're doing research, and we're even testing for COVID. Because
in the stool.
In the stools. That was my main focus during the pandemic, was looking for COVID in the stools, because I said, you know, I'm transplanting patients. What if I'm giving COVID at the same time, right? So to me, it was important to develop an assay. Unfortunately, it's under research assay. It's not an approved test, but at least it gives me confidence that my patient doesn't have COVID, my donor doesn't have COVID. And we test obviously nasal that the FDA requires, but typically.
Yeah, the normal respiratory.
Richard, I think there's a need for looking at the stools, because personally, I've seen so many cases of nasal negative and positive in the stools, you know, and those are your people that have long COVID, that have digestive problems, that are still persisting in having COVID in their gut, creating havoc. So I think, you know, I'm going to have to fight with the FDA to get that test approved, because I really think I don't know how people can treat long COVID without knowing whether you still have COVID in the stools or not, in my opinion.
Just before I ask you, I want another question about COVID, but the fecal transplant can be as simple as getting some poo.
Yes. So it's basically, you want to create, and you want to give it, you want to process the stool to keep it as anaerobic as possible.
All right.
So because I prefer fresh stools, because that, you know, has a better rate of engraftment, in my opinion. And I think, you know, most GI doctors do fecal transplant or intestinal microbiota, like Dr. Alex Karutz likes to say. You know, you still want to have as fresh as possible.
And then it's just like giving an enema. You basically.
Yeah, I mean, I do it via colonoscopy. You know, we basically wash the colon. We make sure, you know, we give if for C. diff, we give them vancomycin before to kill off everything. It's really, think of it as gardening, right? You've got a whole garden full of weeds. You're removing the weeds, and then you're turning the earth, and then you're implanting the seeds, and you put water and sun, and then your fruits are going to grow. So it's the same kind of thing with fecal transplant, where you're basically killing off all the bad stuff, and then you start fresh by cleaning the colon completely, and then implanting a new microbiome, a new fertilizer, let's say, to start implanting into the colon. I mean, that's the best way to kind of, you know, describe it.
So you kind of clean the colon out. The patient presumably doesn't eat for 24 hours. You give them a laxative to clear it out.
Yes, it's exactly like a colonoscopy, and then we go in there and we wash the colon and make sure, and so, you know, you want to make sure that everything is, you know, clean, and that when you're implanting, you're just putting in the microbes so that they could just start growing. Yeah. So obviously,
so you physically wash the colon out during colonoscopy.
Yes.
Is that well tolerated by the patient?
Yeah, I mean, it's, you know, I mean, this is something we do anyways when we do colonoscopies, not to miss polyps. So this is not new in GI. You know, I think it's important to have a good look at the colon, at the colonic mucosa, and it's important to clean the colon before you implant, in my opinion. Yeah,
absolutely. So next question about long COVID then. In your experience, people with long COVID are still secreting active whole virus.
Some people we've discovered have whole virus in their stools, like up to a year after having had COVID. So if you don't take care of that, you're not going to fix the problem. So, and long COVID to me is defined as a couple of ways, right? It's either the virus is still persisting, you've had the virus and you took some medications that killed the virus, but also wiped out your whole microbiome, so you never really come back from that, or you started off with dysbiosis to begin with, got COVID because of the dysbiosis, and then essentially are having a hard time going back to pre-dysbiosis, right? So it's really three different ways of looking at it. And then, of course, you know, if you believe in the whole spike, you know, in the stools as well, possibly, is that causing some problems down the road in the colon? So, you know, I think long COVID is not that straightforward, but to me, it's gut dysbiosis. We've published on that. We did an abstract that we presented last year at ACG, where we showed long COVID patients were lacking certain microbes and had an overgrowth of other microbes. So it is a leaky gut. It is a dysbiosis. Whether it's caused by the virus or caused by the treatment that you took for the virus, that's, you know, who knows? You have to look at history from the patient.
For sure. I mean, I think there's a long COVID and post-vaccine long COVID. I think there's
Yes.
probably two
So
separate syndromes.
Well, it's interesting because I think the important thing to kind of take in the history of the patient, because I have people that come in and say, I have long COVID. And then you take the history and you say, well, when did you have COVID? Oh, I had it in January 2022. When did you get vaccinated? March of 2021. Oh, okay. So you got the vaccine that was supposed to protect you from COVID, but you got COVID.
Yeah.
And therefore, I don't know that that's a long COVID. I think that was the beginning of the demolishing of the spike protein from the vaccine that started that. So, and we'll discuss that later. We discovered that the spike protein kills the bifidobacteria, which is an important microbe.
Oh, right.
And so, when you start taking the history of these patients and you say, well, you had the vaccine, our data showed that you could, up to 9 months later, the spike protein keeps killing the bifidobacteria. So when a patient tells you, well, I got COVID a month after my vaccine, or I got COVID 9 months later, and I kept getting COVID, is that a vaccine side effect, or is that COVID side effect that caused the dysbiosis? I challenge to say that it started off with the vaccine causing the problem. So to me, that's how I differentiate them.
So there could be two pathologies going on here. Presumably, if there's active virus being excreted, what did you say? 9 months later or something?
Yes. So you can have COVID in the stools up to a year later. That's the longest we found.
Right.
I'm sure it's in others, but.
So the virus must be reproducing inside the cells of the gastrointestinal tract somehow.
Yeah, I mean, it does, there's been data in GI that shows that it is persisting. So, it's not just Sabine Hazan saying it. So there's definitely data out there that it is persisting, it is reproducing. Listen, we tend to think of our poop as just poop, but these are live microbes in there, right? We have live microbes that are living in our guts and are doing some function. They're either there to, break down sugars. They're either there to break down calcium. So these microbes are very well alive in your gut. Now, what happens when you introduce a virus to that live environment of microbes? For the most part, it'll start what's called a bacteriophage, killing those bacteria. Remember, a bacteria is 20 times bigger than a virus. So, you've got these big bacteria that are basically getting killed by an overaccumulation of viruses in the gut. It is causing problems, and it is causing what's called a bacteriophage. So, and in the case of bifidobacteria, it's causing a bifidophage.
Right, so in the same way that viruses can infect my cells,
yes,
viruses can infect bacterial cells. So a bacterium can get a viral infection.
A bacterium could basically get killed by a virus.
And that's the bacteriophage.
And yes, and remember, these microbes, interconnect. They're basically sequences of microbes, right? So, is a spike and that was the beginning of my questioning, right? At the beginning of the pandemic, we had an interesting case where we had 2 vaccinated patients. We discovered that they had, and actually Dr. Robert Clancy, who was here in LA, looked at that data and was really, fascinated by that because, and asked the same questions I did, which is, how is it possible that in 2021, it was in July 2021, we discovered the original Wuhan strain in 2 patients that were vaccinated. So remember, by the time the virus came to America, it had already passed from China to Europe to Italy to America, right? When it got to here, it was already, we were 4 or 5 spike mutations in the virus. When you see a virus with 3 parts only, that's the original Wuhan strain. How does that happen? I propose the hypothesis that perhaps the spike protein entered the blood, traveled to the colon, found an old coronavirus from, I had a coronavirus in my gut before the pandemic. You go to bat caves, you're going to be exposed to coronaviruses, right? Now,
there's 7 human coronaviruses anyway.
Correct. So now, is there a coronavirus that is basically activated by that spike protein that was mimicking the Wuhan strain? I mean, if you think back of the whole way, and I think everybody by now realizes, we're in the world of designer bugs, right?
Yeah.
They have the capability to take tuberculosis and a virus and put it together and create these design bugs, right? So it was a design bug. This was a designer bug. This was a creation of sequences, right? That you basically say, I'm going to take A, B, C, and I'm going to join it with D, E, F, and I'm going to create a novel microbe, because now I can patent that novel microbe, and now I can have a treatment that's a vaccine that's A, B, C, D, E, F that matches that novel microbe, and I can license that novel microbe. I mean, this is the world we're living in right now.
It's quite a good business model, really, isn't it? You invent a new bug, then you invent a vaccine to treat it.
Well, because you cannot patent bugs. You cannot patent known microbes. You cannot patent Clostridium difficile, for example.
But you can patent vaccines.
But you can patent a combination of Clostridium difficile with a Clostridium perfringens.
I see. This is worse than I thought. So I knew people were jiggly pokering around with viruses, but they're actually combining viral genetic material and bacterial genetic material.
Yes.
But that's that wouldn't happen in nature normally. It's going to produce something completely new.
And unfortunately, this is the world we're living in, and this is why we need to stop that, because we are creating superbugs, and that's not going to be helpful to fix this problem, right? Because, listen, the body rejects the unnatural, but up to what level? Certain people will survive. They have a resilience in their microbiome. They're resilient. Evolution, the strongest will survive. But how many will die from those experiments, right? And yes, you could do an experiment just in case a microbe does this with another, because that's the reasoning, right? In the mind of a scientist. Well, we better experiment to see what's going on just in case it happens in nature later on. But here's the problem. You're creating the just in case it's going to happen in nature down the road, right? I mean, it's like, I hear scientists saying, oh, wouldn't it be cool if we could reproduce a dinosaur? No, it would not be cool to reproduce a dinosaur, okay? I mean, yeah, if you're, smoking something and you want to see a dinosaur, but the reality is, what is that dinosaur going to do once it starts growing up? It's going to kill humanity. So I think we tend to push these experiments, and there's a lot of scientists in their labs right now doing a lot of crazy stuff, and we need to really have better supervision of all that.
Absolutely.
So, yeah.
Just tell us what a dysbiosis is, Sabine, please.
So dysbiosis is an imbalance of the microbiome. It's, I don't like to think of bugs as, or microbes as good or bad, because I really think they're good up to a certain level, they're bad to a certain level. But you have to imagine that if you have an overgrowth of a microbe that is supposed to break down sugar, and then you have an undergrowth of a microbe, like you've lost the microbe that breaks down sugar. And now you have another microbe that's doing something else, but it's too high. That imbalance between the microbe that's overgrown and the microbe that is useful to brown sugar is really the dysbiosis. We're going to come out with a definition, and that's one of the projects I'm doing with Dr. Clancy, is basically, and also a scientist that used to work at the National Institute of Standards, to basically look at all the data. We have thousands of stool samples of patients with diseases, because we're conducting 61 clinical trials on the microbiome. So my goal, I know. So, you think about, what does the microbiome look like in diabetes? What does the microbiome look like in alopecia areata? What does it look like in, pemphigoid disease. What does it look like in lupus? So all these diseases, we wrote 60. Remember, I'm the girl that was doing clinical trials for pharma. So I have about 300 protocols in my closet, in my storage, from different conditions. So we were able to kind of compile all these protocols and make them specific to the microbiome and certain diseases. And then from there, we basically are trying to map the microbiome and diseases. From there, what we've noticed is that there is something that we call dysbiosis, which is an imbalance of the microbiome. So that's what dysbiosis, leaky gut that you call is. You have to think of your stools as the first wall of protection. When you ingest things that have viruses, fungi, parasites, your stools is really what's your defense, your first defense. Then you've got your colonic mucosa, then you've got your blood vessels, then you've got your nerve. So if your stool, your barrier is gone, those microbes are passing, and you have leaky gut, those microbes are passing from the colon to the blood vessels, to the nerve. They travel, they secrete metabolites, which basically then go to the brain, causes problems. And that's what we're starting to see with Alzheimer's, Parkinson's, MS, ALS, these microbes that are basically out of whack, and metabolites going up and down and causing nerve damage.
So when I was young, the idea was that the gastrointestinal tract was basically this barrier that large molecules like proteins and things couldn't get through. But that's wrong, isn't it? This idea of a leaky gut.
You have to start at the microbes. We trained, and you and I, probably the same generation, we trained with cytology, right? Remember cytology, histology, and we trained with mitochondria and Krebs cycle, and that's how we learned, right? It was all at the cellular level. But before the cellular level are microbes. So before you even get, so I like to, if this is my hand, if this is my colon, right? And I'm ingesting sugar in my gut, and now I have my sugar goes into my colon. There's microbes in that colon that are helping that sugar break down to penetrate, to let it go into the cell. Then what's in the cell, the mitochondria is basically using it. The Krebs cycle is using it. Function of the cell happens. But before all that are the microbes that are doing their job. Now, if you are lacking those microbes that are breaking down your sugar, that are breaking down your calcium, you're never going to put that sugar in the cell. Where's that sugar going? Everywhere else but the cell.
So the dysbiosis can actually affect the wall of the gut, so it becomes leaky. Does that mean things like large molecules and even parts of bacteria can get from the lumen of the gut into the blood and therefore make you feel sick?
Well, that's the concept, right? That's definitely the thought process, the hypothesis. That's why you start seeing people that have chronic UTI. The work of Dr. Sahil Khanna at Mayo Clinic, he did a lot of work looking at people with C. diff that he did fecal transplant, and they had chronic urinary tract infections. And when he restored the gut, he improved the chronic urinary tract infections. Did the chronic urinary tract infections occur because of that leakage, because of that loss, because of those lost microbes, right? We tend to think as microbes as bad, but we also forget that microbes are good. And so when you kill the bad, you're killing the good as well, right? Remember, during the pandemic, what did we do? We were so, we were hunting the virus, and we were all afraid of the virus that we ended up taking all these meds that were killing the virus, but also killing the microbiome. And so, yes, your microbiome will come back, with time and good nutrition, etc. But unfortunately, the damage is done. We think of microbes as bad and causing a problem, but we don't think of the lost microbes causing the problem. That's what I'm trying to really get all the doctors to start thinking of. Guys, stop killing. You've killed enough. You think of all these. I give lectures at the ILADS, Lyme Disease Association, right? And I speak to all these doctors I'm the missing link, right? Because everybody, kills at those meetings to basically kill the bug, right? Kill the tick. And they forget you need to replenish the gut. You need to start focusing on healing the gut because you've just killed too much. And in time, we're going to have better diagnostic tools for Lyme disease where we could say, okay, you know what? We've killed the bug. Now we got to replenish the microbiome. You know, it's kind of like C. diff. We give vancomycin for 10 days. We replenish with a new microbiome, and then we let it be. We don't see these patients ever again. So
if I wanted to get a dysbiosis, I would take antibiotics.
that's one way. You could get a dysbiosis by drinking a lot of alcohol. You can get a dysbiosis by taking on a lot of products out there that are actually, you know, not as good for your microbiome. You know, there's so many things out there from, you know, in the nutraceutical world even that can kill the microbiome. Think about the preservatives that they put in all these products to preserve them. Is that killing your microbiome? Think about the capsules. What kind of capsules are you putting your vitamins in, right? So we were the lab that showed vitamin C improves the bifidobacteria, the good bacteria. If you have a capsule that is of a certain substance that kills the bifidobacteria, it doesn't matter that you're giving it vitamin C because you're now killing it with the capsule itself.
So the material the capsule is made from
The material that you're making the capsules from is important. The contaminants is important, can kill the microbiome. Like I said, alcohol, high consumptions of alcohol, certain drugs kill the microbiome. You know, certain products kill the microbiome. So there's a lot out there, and we're just kind of tapping the surface of what kills and what heals. And I think as we get into this world of the microbiome, it's important. And that's really my work, is really to see what is Manuka honey doing in the microbiome? What is apple cider vinegar doing to the microbiome? What is cumin doing to the microbiome? I think we really need to start looking. You know, it's one thing to look at mice studies and animal studies, but they unfortunately do not reproduce in humans. We really need more human studies on all these natural products to really, because here's the problem. When I'm treating my patients, I'm trying to regrow microbes. If I give it something else that destroys the microbiome, it's not going to help. Absolutely. You know, it's like putting water on a fire and gasoline at the same time. You're never going to turn off your fire.
Trouble is, there's not much money in honey, is there, really?
No, there's not much money, but we don't know what honey is doing. Actually, I'm excited because we're doing a study on honey.
Or apple cider vinegar. I mean, what I'm saying is these things can't be patented, so there's no impetus to put big pharma money into it.
Well, but I'm doing it, so I don't care. I want to know.
So do I. Thank you.
I spend more money on stool studies than anything because I have a curious mind and I want to know. And ultimately, like I said, I cannot reach success. And we're gonna talk about a certain, bunch of cases that I'm treating by manipulating the microbiome. I cannot reach success if I don't know these things, if I don't know what all these drugs are doing on the microbiome. There is a model that was done. I'm excited because I'm working with this new scientist now, and he told me about this colon model that Harvard developed, where you basically don't have to wait to enroll patients. It's kind of like an in vitro model, where you basically give the product and see what it's doing to the colon, in essence. It's a colonic model. So I'm excited to work with that because it's going to speed up the research to do the in vitro study first. Because think about what I do now. I'm taking my patients, I'm giving them the same exact protocol of food, and I'm changing, I'm giving them a lot of Manuka honey after a week and then retesting, right? So it takes a month to get those results, and then it takes another 6 months to a year to gather at least 20 patients. It's much faster to do it in vitro to begin with.
But presumably Californian honey is pretty good as well, is it?
Yeah, I mean, it should be, hopefully. We have a lot of problems here. We have fires, we have mudslides.
Yeah.
So I don't know. That's I do have 10 beehives, so I can tell
Okay.
you I have my own honey. Yes.
Fantastic.
I'm all about protecting the bees.
Oh, absolutely. Yeah, we won't start on that one. We agree on that. But how
Yes.
much vitamin C do you take personally?
Well, during the pandemic, I was taking between 1,000 to 3,000 milligrams.
Right.
I don't take anything anymore. I've kind of like, balanced my microbiome. I'm a little bit privileged in that I have a lab that can test. So I try to,
Yeah.
I always see, okay, I'm engrafted. I've got some good bifidobacteria. I'm good. I don't need to worry about it. And it's also summer. So you remember you're outdoors, you're exposed to microbes, you're essentially it's much better, and we're getting vitamin D from the sun, which also, by the way, increases bifidobacteria. So
Oh, does it? Right.
yeah, so you don't really
We-
need as much.
Maybe in California you get vitamin D from the sun. We don't get too much.
Oh yeah, where you are, yes.
Yeah.
So where you are, probably increase the vitamin C, yes.
Yeah,
But
and of
between
course,
1,000 to
the
3,000.
older you get, the less your skin makes vitamin D, of course. So,
I think the older we get, the number one vitamin people need is vitamin D and
Absolutely.
K2, of course.
Absolutely, yeah,
Yes.
absolutely. So you're giving these fecal transplants. Let me get this right. That is a probiotic, isn't it?
I guess you could think of it that way. Probiotic is really a couple of microbes, the bifidobacteria, the lactobacillus. Fecal
Right.
transplant is really a consortium. It's like 10,000, 15,000 microbes that we've identified that we put in. It's literally the stool